What happens inside a mother's womb during pregnancy is a source of wonder and mystery for many. Embryology is the branch of science that aims to unlock these secrets. It is the study of the development of an embryo, exploring the intricate processes that transform a single fertilized egg into a complex living being. From the moment of conception to the moment of birth, embryology unravels the remarkable journey of life in its earliest stages. Think of embryology like assembling a complex puzzle. Each piece, each stage of development is crucial to the formation of the final picture, a healthy baby. By understanding how these pieces fit together, we gain a deeper appreciation for the miracle of life itself. This field of study delves into the fascinating world of cells, genes, and the intricate signals that orchestrate this incredible transformation. It's a journey filled with scientific wonder and a testament to the remarkable power of nature. Why is it important to understand embryology? The answer lies in the profound impact this field has on various aspects of human health and well-being. For medical professionals, embryology is not just an academic pursuit. It's a fundamental pillar of their understanding of human development and congenital anomalies. By studying the normal development of an embryo, doctors and scientists can better understand what happens when things go wrong. Birth defects, for example, are often the result of errors during embryonic development. Knowledge of embryology helps in diagnosing, preventing, and potentially even treating these conditions. Furthermore, embryology plays a vital role in reproductive medicine. In vitro fertilization, prenatal screening, and gene therapy all rely heavily on the principles of embryology. Understanding how embryos develop allows us to develop new technologies and therapies to assist couples struggling with infertility and ensure the health of future generations. The journey of human development begins with a single fertilized egg, known as a zygote. This microscopic sphere of life embarks on a remarkable transformation, dividing and differentiating into trillions of specialized cells that make up a human being. The first stage, called cleavage, involves rapid cell division without significant growth. The zygote divides into 2 cells, then 4, 8, 16, and so on, forming a cluster of cells called a morula. As the morula continues to divide, a fluid-filled cavity forms within it, transforming it into a blastocyst. This hollow ball of cells marks a crucial stage because it's during this time that the embryo implants itself into the lining of the uterus, marking the beginning of pregnancy. Around the 3rd week after fertilization, a process called gastrulation begins. During gastrulation, the cells of the blastocyst reorganize into 3 distinct layers called germ layers: the ectoderm, mesoderm, and endoderm. These layers will eventually give rise to all the different tissues and organs in the body. The ectoderm forms the skin and nervous system, the mesoderm develops into muscles, bones, and the circulatory system, and the endoderm gives rise to the digestive system and lungs. Following gastrulation, the embryo enters a period of rapid growth and development known as organogenesis. This is when the rudimentary organs begin to form from the 3 germ layers. The heart, one of the first organs to develop, starts beating around week 4. By the end of the 8th week, most of the major organ systems are established, and the embryo is now considered a fetus. Let's take a closer look at some of the key players in this incredible journey of development: fertilization. This marks the very beginning, the moment when a sperm cell successfully penetrates an egg cell, fusing their genetic material to create a zygote. This union is the spark that ignites the entire process of development. Blastocyst. Imagine a tiny fluid-filled balloon. That's what a blastocyst looks like under a microscope. This hollow structure is not only important for implantation, but also contains the inner cell mass, which will eventually develop into the fetus. Gastrulation. This is a critical turning point in development. Think of it like a well-choreographed dance where cells move and rearrange themselves to form the 3 primary germ layers, laying the foundation for the development of all tissues and organs. Organogenesis. This is the stage where things really start to take shape, literally. From the primitive structures formed during gastrulation, specific organs begin to develop. It's a period of rapid growth and differentiation, transforming the embryo into a recognizable miniature human being. 2 fundamental processes drive the remarkable transformation from a single cell to a complex organism: genetics and cell differentiation. Genes, inherited from both parents, provide the blueprint for development. They contain instructions that determine an individual's traits, from eye color to susceptibility to certain diseases. Cell differentiation is equally important. During development, cells don't just multiply, they also specialize. A single fertilized egg gives rise to hundreds of different types of cells, each with a specific function. Some cells become skin cells, others become nerve cells, and so on. This specialization is guided by a complex interplay of signals, both from within the cell itself and from its surrounding environment. Imagine a group of students with the same textbook, genes. Each student can choose to focus on different chapters and specialize in a particular subject, cell differentiation, leading to a diverse range of professions and skills. That's essentially what happens during embryonic development, but on a cellular level. Our understanding of embryology has revolutionized various fields of medicine. Infertility treatments, prenatal diagnosis, and regenerative medicine have all benefited immensely from the knowledge gained through embryological research. In vitro fertilization, or IVF for example, relies heavily on embryological principles. By understanding the intricate processes of fertilization and early embryonic development, scientists and clinicians can assist couples who are unable to conceive naturally. Prenatal diagnosis, another area significantly impacted by embryology, allows doctors to detect potential birth defects early in pregnancy. Techniques like amniocentesis and chorionic villus sampling enable the analysis of fetal cells, providing valuable information about the developing baby's health. Furthermore, the field of regenerative medicine holds immense promise for treating a wide range of diseases and injuries. Stem cell therapy, a rapidly evolving field within regenerative medicine, relies on the principles of embryology to grow new tissues and organs. The study of human embryos, while offering incredible potential for medical advancements, also raises ethical considerations. The use of embryos in research, particularly those created through IVF, is a subject of ongoing debate. Different societies and cultures have varying ethical and legal frameworks governing embryological research. One of the main ethical concerns revolves around the moral status of the embryo. When does an embryo acquire the same rights and protections as a born human being? This question is complex and doesn't have a simple answer. Different perspectives exist, ranging from those who believe that life begins at conception to those who argue that an embryo doesn't attain full moral status until a later stage of development. Balancing the potential benefits of embryological research with ethical considerations requires careful thought and ongoing dialogue. Open communication between scientists, ethicists, policymakers, and the public is essential to ensure that research in this field is conducted responsibly and ethically. Embryology is a dynamic and rapidly evolving field, with new discoveries constantly reshaping our understanding of the earliest stages of life. Advances in imaging techniques, genetic sequencing, and stem cell research are opening up new frontiers in embryology, promising exciting possibilities for the future. One area of active research is understanding the molecular mechanisms that govern cell fate decisions during development. By deciphering the intricate signaling pathways that determine whether a cell becomes a neuron, a muscle cell, or a blood cell, scientists hope to develop new therapies for a wide range of diseases. Another promising area is the development of artificial embryos or embryoids. Scientists are now able to create embryo-like structures from stem cells, offering a potential alternative to using embryos derived from IVF for research purposes. These embryoids could provide valuable insights into early human development without the ethical concerns associated with using actual embryos. The journey from a single fertilized egg to a fully formed human being is nothing short of miraculous. Embryology, the study of this incredible transformation, provides a window into the very essence of life, revealing the intricate processes that shape us from our earliest beginnings. Understanding embryology is not just about satisfying scientific curiosity. It has profound implications for human health, medical advancements, and our understanding of ourselves. As we delve deeper into the mysteries of early human development, we gain a greater appreciation for the complexity and beauty of life itself. The knowledge gained through embryological research holds the key to unlocking new treatments for diseases, preventing birth defects, and ensuring the health of future generations.